Hello, everyone. I'll go ahead and get started. I believe that everybody is joined today. And I'm Hikmet Budak. I'm the IWGSC board member. And I'm joined today by Scott Boden. And he is going to be talking with us today is about the finding new baby gene in wheat. And before I go ahead and get started with the presentation, I just wanted to give you a quick overview about the IWGSC and to remind some of the information. And we have a non-governmental organization, and with the over 3,400 members, and then over 900 institutes and companies in 72 countries engaged with us. So we do have, as an international wheat genome sequencing consortium, that help with the sponsors, and without their help, we couldn't have coordinating and organizing this webinar and all the genome-related efforts without their support. So I'll go ahead and take a quick moment to acknowledge each one of IWGSC sponsors: Ann Arbor Bioscience and BASF, CIMMYT, Florimond Depros, INRAE, Illumina, Kansas Wheat, RAGT, and Syngenta. Again, thanks for all the supporters and sponsors. And after we published the reference sequencing in 2018, our vision is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity, and then to develop, and that has included revising the version and as well as the functional and structural annotation of the wheat genes and genome are the main goal, to develop tools and so that everybody can use it. And before I go ahead and continue, that I just wanted to announce that our next webinar is going to be presented by Andrea Gonzalez from King Abdullah University of Science and Technology in Saudi Arabia. And he will be talking with us in July 13, is that the Agilep's Tasha pen genome and a tool for harnessing the wild wheat. And the registration will be announced and then distributed. And this is the link for the registration as well. Just a quick reminder about the webinar dashboard, and the webinar is going to be recorded and will be posted on our IWGSC YouTube channel. And please just subscribe to the channel so never miss a new upload as well. In the webinar dashboard, that you will see that 3 main boxes, and then one of them is going to be questions. And please just submit your questions and to the question panel, and we'll try our best to answer all the questions. But if you cannot answer all the questions, Scott agreed to just answer the question after the webinar by email. And please just check the chat box as well, monitor that we will send some information regarding this presentation, and as well as the next presentation and papers too. And then handout dashboard, you can download the Scott's presentation as well as that our the introduction as well, and then the papers PDF version too. Again, I'd like to welcome Scott Boden, and Scott is the group leader at IRC and also ARC Future Fellow at the University of Adelaide, Australia. And then he will be talking with us today is the finding a name for your new baby wheat gene. And I'd like to welcome Scott and as well as the old participant as well. Thank you so much, Scott. This is all yours.
Great, thanks very much for the introduction. There we go. Okay, so Yeah, thanks very much for the introduction. It's a pleasure to be here with you all today. I thought it would be important to introduce myself to begin with, given that there's probably a lot of you that I haven't yet had the pleasure of meeting. And I realize that today's talk could be a little bit controversial, given that we can see in the literature that there are many different ways in which people decide to name genes, not only in wheat, but in other organisms as well. And so this is a topic that I've had quite a long-standing interest in. I've been working on wheat for nearly 20 years now, and I've been working on topics including meiosis and flowering time and spike development and grain production. And during that time, I've been trying to discover genes that regulate processes that occur during each of these particular events of the plant's life cycle. And so during my time traveling around the world, within Australia and the UK and Japan and America, I've been talking to a number of different researchers about how genes are named in wheat. And during this time, I've developed an appreciation that has been quite a history to naming genes or genetic elements in wheat. So from naming the different genomes to naming chromosomes and QTLs, there's been quite a long history to this. And that's something that I've appreciated through talking to people like Bob McIntosh, who's one of the authors on this paper and has been curating the wheat gene catalogue for quite a few years now. Talking to Peter Langridge, who was one of my supervisors, and Takashi Endo and Graham Moore, but also through reading the works of people like Ernie Sears, who did a lot of the sort of fundamental genetic work in wheat. And I really think that now is the right time that we update or refresh these guidelines, because there's been quite an explosion of information about wheat genetics in the last decade or so, particularly with the sequence information for the reference genome. And this has really accelerated the rate at which genes have been discovered in wheat. And so we've seen many more papers coming out talking about genes in wheat. And I think it's really important, especially for that objective of the IWGSC, to try and understand how we could use genetics to enhance the breeding potential of wheat, that we have a good system for naming these genes. And so that's kind of what's led to this international and multidisciplinary effort to try and update the guidelines for wheat gene nomenclature. And I want to emphasize with this that this was very much a collective effort. And while I'm the speaker today, this was very much a collaboration between many different researchers from different countries and trying to span many different disciplines where gene nomenclature has been quite important in the last decade. And one of the things that I really like to encourage you today as listeners to today's talk is that you go away from today's talk and you perhaps discuss it with your colleagues, and you go and read the manuscripts and the guidelines that we've published to try and understand the best method for naming genes in wheat. And that this is, I guess one of the messages that I want to have from today is that this isn't going to be a static model by which we name genes. We appreciate the fact that there's going to be new technologies coming on board, and there's going to be new information coming on board as we sequence more and more cultivars. And so this needs to be sort of a flexible model, and that can really only adapt in the best possible way if we have input from you, from the wheat research community. And so my role within these new guidelines has really been just through starting the conversation. So as we perhaps all appreciated during COVID, there was time for reflection and for thought. And while reviewing quite a few papers, I realized that there was different methods being used by researchers to name genes in wheat. And so this is an example of a Twitter conversation that I started back in 2020, which stimulated quite a lot of interest within the research community. And one of the people that responded was Christabel Wowie at the John Innes Centre. And through this conversation, we realized that the accessibility to the guidelines that were then current for naming genes in wheat wasn't so great. So it was hard to find these guidelines on the internet. It was hard to understand the way in which genes should be properly named. And so we thought that that was contributing to a little bit about the confusion on how genes should be named. And so we thought that this was really the right time to try and update those guidelines and give people better accessibility to those guidelines. And so just to give you one example of a gene that we noticed was being sort of misnamed or was being named differently in different papers, and how that was potentially leading to confusion, was a gene that I'm quite familiar with, which is called VernA1 or vernalization A1. And vernalization A1 is a gene that controls the spring growth habit in wheat. And it's a gene that needs to be induced to promote flowering in response to the cold treatment in winter wheat. And what I noticed was that this gene was being named in a few different ways. So it was either being given the name VernA1, it was being called Vern 5A because the gene is located on chromosome 5A, or Vern1 5A. It was being referred to based on homology in other species, such as rice, where it was being called MADS14, or in Arabidopsis, where it was being called AP1A1 or AP1 5A, again representing where its chromosomal location. And so I could appreciate how this would be quite confusing. And what I noticed in quite a few papers was that there was incorrect citation of this gene and where it had been discovered, because people hadn't realized that it actually had a different name, and that had been named in a number of other papers in a different way. And so if we think about all of these different names by which it's been called, there's actually two versions which is the correct name, and that's one of the things that I'd like to explain in this talk, is how this gene could be named VernA1, but also be named in a different way, AP1A1, and how these are both correct. So I've sort of already touched on this, but the reason why we wanted to update the guidelines is because there was already a set of guidelines that have been published, and these were curated by Bob McIntosh, and these existing set of guidelines formed the basis of this updated version. And as I mentioned, we found it hard to find and it lacked information. So Christabel and I both went to go and search for these existing set of guidelines, and we found that the accessibility wasn't so great, potentially because it had been uploaded onto websites which were no longer maintained, and so they were difficult to find. But we also realized that when we did find these guidelines, that there were a number of areas related to modern day genetics and genomics that weren't covered in these guidelines. So we're in reference to the genome sequence data that was available for wheat, and the technical advances that have come about through improved transformation methods, gene editing, the development of markers based on SNPs, and the development of new resources such as tilling populations. These weren't covered within those guidelines, and so we thought that it was best that we update them to include these new advancements. We also appreciated that there'd been an expansion of the wheat research community. So there were more genes that were being discovered, there were more researchers investigating wheat, and what we assumed from reading a lot of these papers is that there were people coming into wheat from other organisms who were bringing the genetic knowledge from those other species into wheat, and they were naming genes accordingly based on what they'd researched in other organisms such as rice or maize or Arabidopsis. And that's fantastic. Like, we definitely welcome new people coming into the wheat research community, and the more genes that are discovered, then the more possibilities we have for improving breeding. But we noticed that this was coming with some potential confusion with gene nomenclature. So what do we hope comes from these improved or these updated guidelines? So hopefully there'll be improved clarity on how to name a gene in wheat. And hopefully, if we all agree on these guidelines, then it will be a model by which we can all follow when we come to publish papers or name genes and pass them on to breeders. We hope that there will be reduced complexity in the literature. So one thing that I've noticed is that for a set of genes that I've been working on, which are MADS-POTS transcription factors, such as VernA1, there's been incorrect citations, and there's sort of been a lack of transfer of knowledge between research groups because genes have been named in different ways. And so what we hope from this is that there'll be better transfer of knowledge among the wheat research community, and this will help accelerate our discoveries, not only of the genes, but also the biological processes that these genes were involved in. And what we hope is that this system could adapt to advances in genome sequencing and technical capabilities from now into the future. it could provide a reference not only for the academic community, but also for the breeding community. So when we put this question out there about how genes are named, we found out that the breeders actually like genes being named based on their chromosomal location because it helps with the breeding process. But that comes with potential confusions, as I'll explain a little bit later. And so hopefully these updated guidelines will provide a good reference for both the academic and breeding community. And as we learn more about other crops that are related to other temperate cereals that are related to wheat potentially, these guidelines could be used by those other crops. So barley and oats and rye are immediate examples that come to mind. Following on from these reasons, I guess why I'm encouraging you all to follow these guidelines is that as you have invested a lot of energy into discovering a new gene, you want to get the most impact from that discovery. So when you find your new baby gene and you want to put it out there into the world, and you want to give it a name, and you want people to see your research and cite it, then having improved clarity on how to name that gene, I think will definitely help with the impact of your research. Reduced complexity in the literature will mean that following on from this, your work will have bigger and broader impact, and hopefully will get cited more often. It'll be a set of guidelines that will make it easier for the academic and breeding communities to communicate with each other and also to transfer knowledge. And we also think that there would be potential for this to transfer in knowledge into other cereal crops. So one of the things that I've noticed as being a wheat researcher is that we're often naming genes based on genes that have been discovered in wheat, in rice or maize or Arabidopsis. Maybe this will give us an opportunity we get to name genes first in wheat, and then some of the other cereals will be naming their genes after our own discoveries in wheat. So just as a bit of a disclaimer, again, I recognize that this is potentially a controversial topic. This is something where a lot of people will have different opinions. And this indeed came out while we were discussing the guidelines with the collective authors for this particular manuscript, is that there were different opinions, and we tried our best to try and collate those different opinions and come up with a consensus. But we appreciate that you will have some different opinions about how genes should be named. And so these guidelines are not meant to be too prescriptive. We're not out there to be police to try and tell you how you should name your gene, but we appreciate that when you discover a gene, you might not know where to start. You might not know what the best way is to name a gene. And given that there is some differences out there in the literature, you might be following the wrong example. And so we hope that this would just give you a common reference point without wanting to tell you exactly how you should be doing it. We understand that there will be exceptions to the rules, and we're happy for the guidelines to adapt to new genetic and genomic advances. So as new technologies come on board, these guidelines will have to adapt to needs to name things according to those new advances. And we understand that there will be different reasons why you might need to sort of bend the rules a little bit with these guidelines, and that's perfectly fine. Again, if you sort of use this as a common reference point, then hopefully those names won't be too different from what everyone else is using. As I said, you won't be policed. We're not out here to tell you that you're doing the wrong thing. And I myself put my hand up and say that I've named genes in my publications in a method that's not consistent with these guidelines in the past. And so hopefully, for my own research, this will provide a good reference point by which I can name genes. And we think this is particularly important in the publication of scientific journals. And so we encourage you as reviewers for other people's papers, and also if you're an editor for a scientific journal, to refer people to these guidelines if you notice that they're not naming genes that's consistent with them. We hope that it will promote discussion, and we encourage this to share with your colleagues, especially those who are new to wheat research. So again, it's fantastic that people are coming into wheat research from rice and Arabidopsis and deciding to do their genetic work in wheat. We would like to enlighten them to the ways of wheat research, and so we encourage you to use these guidelines as a way to sort of share knowledge about gene nomenclature with them. And one of the reasons that we include a lot of international authors is that we wanted to sort of encourage an international discussion about this. So if you notice someone who's on the author list who's from your country and please go and talk to them. They're as equally authors as I am, or Christabel or Bob are, and we'd like you to go and talk to them and discuss gene nomenclature with them. So just getting into the guidelines now. As an overview, we sort of have these different roadmaps in the manuscript to explain how you might go through naming a particular gene. And I just want to use this overview to explain that there's two basic considerations for how you might name your gene based on how your research is, your research is occurring. So, and those two different processes are through naming a gene based on a morphological or phenotype, phenotypic feature. And the other is by studying an underlying gene that's been identified, potentially through clonal QTL, or because you've identified a gene in another species that you want to study in wheat, and you want to name that gene accordingly in wheat based on how it's named in another species. And so some examples of morphological or phenotypic features by which genes have been named examples include RHT1 for reduced height 1, vernalization 1 for or VERN1 for vernalization 1, and PH1 for pairing homologous 1, all of which have been important genes in the history of wheat genetic research. And what's interesting is that these genes named based on the underlying gene that underpins these particular loci have different names based on how they've been discovered in other species. So RHT1, the gene is DELA and or gibberellin insensitive, and this gene is located on the D genome, so we'd call it DELA D1. For VERN1, the underlying gene is an apoptyla-1-like transcription factor, and so we would call this AP1A1. And for PH1, the underlying gene is a ZIP4 protein that's involved in recombination repair. And this is a sort of an additional copy of ZIP4 that's located on chromosome 5b, and so it's called ZIP4B2. Okay, so just going to the genes named based on phenotype. So an example of this is vernalization 1, and what we're saying here is that we ask you to name the gene based on the principal characteristic of that trait that you're investigating. So if you're investigating vernalization, then you call it vernalization. If you're investigating height, then you might call it height something. If you're investigating root growth, you might call it root growth something. But name it based on the principal characteristic of the trait that you're investigating. The acronym for that gene should contain 2 to 4 letters, and preferably 3. So through my own research, I wanted to give a gene a name with an acronym that had 4 letters, and I was told that I can't. But actually, through this process, we discussed how many letters can be used, and you can use 2 to 4 letters. And we recognize that there are some classic examples within wheat research that have single letters. So examples like include the Q gene, which controls threshability and spike compactness in wheat. But from here on in, we ask that you don't give an acronym with only a single letter. The locus name should be written uppercase italics, and the loci should be named according to their order of discovery. So if, for example, you were to do an investigation of loci that are involved in vernalization response, and you found that there were 3 loci, which is the case in wheat, then you would order those loci with a successively increasing number in ascending order based on the order in which they were discovered. So the first locus would be called VERN1, the second locus would be called VERN2, and the third locus would be called VERN3, etc. And those numbers would be separated by a hyphen. The numbers are based on the order of discovery, as I said, and not based on their chromosomal location. So I guess this is one particular point that we'd like to make here, is that you could potentially start to see how these use of numbers could be quite confusing if, for example, VERN3, which is the third locus that was identified for vernalization, also happened to be, it's actually located on chromosome 7. So if we named it VERN7A because it was located on chromosome 7, then that might become confusing as to how it was discovered and what the actual gene is. And so it's important that we sort of follow this convention so that we don't start to get confused. And another example of this is that there's actually a fourth locus for vernalization. And so if we started to name them 1, 2, 3, 4, then people would get confused as to whether or not they were different genes or whether they're located on different chromosomes. And for homologs of the same set, we ask that you put the genome identifier before the particular number for that locus. So, for example, it should be Vern A1, Vern B1, and Vern D1, and not Vern 1A or Vern 1B, for example. Yeah, and in particular, I just want to emphasize this point that we're advising that you do not include a reference to the chromosome group in the gene name, because this just brings about confusion. If we then begin to talk about alleles for these genes that are identified by phenotype, then dominant alleles will begin with the capital letter and recessive alleles will be all lowercase. And this follows the convention as it is in a number of different organisms. So for example, the dominant allele for Vern1 is Vern1 with the capital V, and the recessive alleles for Vern1 are all lowercase, VRN. Independent alleles that confer different effects are designated with a lowercase letter after the number. So for example, Vern A1A and Vern A1B are different alleles that confer different spring or winter growth habits, and they're separated from each other by this lowercase letter after the 1. As part of this, we also discussed how we might consider heterozygous genotypes, because in animal literature, for example, when we're talking about genetics, then the plus sign is often used for designating a wild type genotype. And it was stated in the original guidelines that the plus sign should not be used. And so what we recommend if you want to write about a heterozygous genotype within your publication is that you describe the different alleles for the particular gene that you're talking about. So you list both of the different alleles and they're separated from each other by this slash. So for example, a spring winter heterozygous genotype would be Vern A1 / Vern A1 with either a capital or a lowercase v. Some other particular notes that come out through this is that we also considered how inhibitors or suppressors or enhancers are described within the literature, and there are some particular nomenclature guidelines that we recommend for describing inhibitors and suppressors and enhancers. So EN is for an enhancer, I is for an inhibitor, and SU is for a suppressor. For chromosomal aberrations such as deficiencies or duplications, inversions, translocations, and transpositions, there are similarly some different notations that we recommend for those particular chromosome aberrations. And for extra chromosomal units for describing genetic elements that are within mitochondria or plastids or chloroplasts, similarly, there are some prefixes for gene names for those particular units. So now if we move on to how we would name a gene that's been identified or a gene that's investigated based on its similarity with other species. So an example for this might be if you map-based clone a gene that underlies a QTL, or if you're particularly interested in a gene in another species, you want to study its effect in wheat, then these are the particular guidelines by which we recommend for those cases. So again, this is like a roadmap where we'd have like a sequence gene based on evidence within the reference genome, and for that there would be a particular a gene ID or a transcript ID, and then we'd be naming the gene based on known functions in other species. So in the particular case of Vern A1, the gene was homologous to a gene of known function in Arabidopsis, which was called apitala1, and so Vern A1 is an apitala1-like transcription factor. And then later on, I'll be talking about particular haplotypes and alleles for these genes. Okay, so we have this locus name, which is Vern A1 in this particular example. And when we cloned the gene that underlies the QTL, we found out that it's an AP1-like transcription factor, and the gene name for the copy on the A genome is AP1A1. And the reason why we have this extra designation of it being AP1A1 instead of just APA1, for example, is because once we started to do some phylogeny, we realized that there were some other copies of Apatala1 genes in wheat. So once the gene's been discovered, in the first instance, we recommend that you can name it with the genus and species designation after the gene name. So for red wheat, that would be Triticum A. AP1A1 or Triticum A. Vern A1. But after the first instance in which it's described, or when it's used in other sections of the paper, we recommend that you drop the TA because it just becomes cumbersome. A really important thing that we also noticed in the literature is that people weren't including the transcript IDs or the gene IDs very clearly within the paper. And so as part of these guidelines, what we recommend is that when you first name the gene in your particular paper, that you include in brackets, for example, the gene ID or the transcript ID for that particular gene. So it is very clear what gene you're talking about in regards to the reference genome. Given that a QTL or a particular locus might be described and has become quite familiar within the literature, such as in this example, Vern A1, we are leaving it up to you to decide whether or not you call the gene Vern A1 or AP1A1 throughout your paper and also in successive papers. So that these names can become interchangeable with each other. And normally what happens is that the literature sort of decides on one and sticks with it, and that becomes the accepted name within the field. And as I said, for this particular example, there are other copies of AP1-like genes in the wheat genome. And so for those that are located on the A genome, these are being called AP1A2 and AP1A3. And then when we talk about the allele names, they can maintain that same convention as what we talked about for Vern A1, where it would just adopt the lowercase letter after the gene name and the number. And then the protein would again follow the same names, but the name would be unitalicized. So if we then come to genes that haven't yet been discovered in wheat, but are based on homology with other species. So this particular example is CCA1, which is an Arabidopsis gene that's involved in the circadian clock. And the wheat gene is also CCA1. And so when we first introduced that gene, we would call it TA-CCA1. And we would call the three wheat homologs CCA-A1, CCA-B1, and CCA-D1. And what's quite interesting is that this is an example of a gene that has been named in the literature, it has been published, and it's been called CCA1-7A and CCA1-7B and 7D, for example, because the gene is located on chromosome group 7. And so this is an example of what we're recommending from now on, that we don't follow that particular nomenclature. We just name it based on the gene name, not based on where it's located within the genome. An example of a gene where there are multiple homologs in wheat, and we're studying that gene because we're interested in its function in wheat based on roles that it has in other species, an example of that is this Sepalata1-like gene, or SEP1. So Sepalata1 in Arabidopsis is part of the ABCDE model for regulating floral organs. And this is a gene that's become of interest in wheat as well. And in wheat, there are 6 different copies of Sepalata1-like genes, and these have been named SEP1-1, SEP1-2, SEP1-3 through to SEP1-6. And each of the 3 wheat homologs would be SEP1-A1, B1, D1, and so forth. One of the reasons why I wanted to sort of raise this as a particular example is because we should come up with, hopefully within literature from now on, we'll come up with a particular consensus about how the gene should be named and stick with that, because this is a really good example of where there have been a number of papers published about the same gene, and there hasn't been very good flow of knowledge between those papers, and indeed they haven't cited each other because the genes are being given different names. So this SEP1-6 gene, for example, is a gene that's quite highly expressed within developing spikes, and it's regulated by a number of genes involved in the photoperiod-dependent flowering pathway. but it's been given different names. So CEP1-6 is also being called agamus-like gene 1 or AGLG1. It's also been called MADS34 based on a gene name in rice, and it's also been called PAP2 or panicle phytoma 2 based on a gene name in rice. And so, through reviewing papers, and this is in my particular area of interest, I realized that there's been a lot of work that hasn't been correctly cited because the genes have been named differently. And so I really encourage us to sort of agree on a particular gene name and make sure that we do our proper research so that we can make sure that we're sort of capturing all the knowledge that's available for our gene of interest. And another example of a gene that I've worked on and reported is a gene called TB1 for teosinte branched 1. And we had a sort of particular decision to make with this because the gene had been named in 3 different ways in 3 different species. So it had been named branched 1 in Arabidopsis, fine culm 1 in rice, and perhaps more famously, teosinte branched 1 in maize. And so, we wanted to name it based on the name that it had been given in another species to try and put it within the context of those other organisms. And we chose TB1 because the phenotypes align strongly with those that have been discovered in maize. The allele had quite a complementary effect to the overexpression that results in the sort of single culm maize plant. And we also wanted to sort of connect with the literature in maize. And so, and we explained all of those reasons in the paper that we published. And I think that was the important thing, is that we were quite open with the reasons why we gave the gene that particular name. And we also provided, all the details, including the gene ID in that paper. Okay, so as a little side note to this, we appreciate that through the study of the pangenome that the Chinese spring reference genome doesn't have all of the genes that also exist within other species. And so if your gene is not within the reference genome sequence, but you found it in another cultivar, so the example here is mace, then we ask that you please provide that specific gene identifier within your paper rather than the Chinese spring identification. And if the sequence is altogether novel, then we ask that you please deposit the sequence into the International Sequence Database collaboration website, which is issued here, and include the accession number in your publication. Again, just to make it as open as possible and to ensure that there's good flow of knowledge between papers. Another sort of separate element which sort of links to the idea of making this relevant for breeders is that DNA-seq is the idea of haplotypes and naming haplotypes. So DNA sequences, haplotypes are DNA sequences that include regions up and downstream of the gene that are of unspecified length. And we ask that in your papers, you include the length of these sequence limits for which you're reporting the particular haplotype. In terms of designation, we recommend that you designate this particular haplotype with the gene name followed by an underscore, a lowercase h, which is in italics for haplotype, and then a sequential number based on their discovery, which is represented here by an X. So as an example for a gene that's been named based on its discovery in another organism, so this is a brasinoid in steroids, brasinosteroid insensitive gene. So we'd name it TA for Triticum Stephen, BRI for the brasinoid insensitive gene, A1 because it's on the A genome and it's the first copy of that gene, and then underscore haplotype 1. Conversely, for a gene that's been discovered based on phenotype, with the example here being RHT1, then it would be named RHT-A1A for the allele in that particular situation, underscore H1, and so forth. One thing that we wish to state about this is that these will not be maintained by the Wheat Gene Catalogue. And so it's really up to you as the author of your particular paper or in reporting to breeders that you sort of recognize how different haplotypes have been named already before you, and that you sort of follow a sequential number in your publication after that. So for example, if 3 haplotypes of RHTA1 had already been reported about, then you start your numbering at number 4. And we really think this is important for not only the academic community, but also the breeding community, especially because breeders tend to rely on haplotypes when making their crosses or informing them what crosses to make. In terms of alleles, this was a really important area of discussion, and for alleles that are based on phenotype, we want to point out that they can be based on plant-based phenotypes, so differences in height, for example, for the RHT alleles, or they can be based on molecular phenotypes. And an example of that might be an allele which confers a different expression of a gene relative to the reference allele. Then you can name it, you can give it an allele designation based on that molecular phenotype. We recommend that you don't give an allele designation to a mutation that's synonymous, only those that are responsible for sort of changing amino acids or changing the expression of a gene, but not something that ends up being a silent mutation. The exception to this would be a mutation within a microRNA binding site. So an example of this is the Q gene, which has a mutation within the microRNA binding site of the apoptyla-2 transcription factor. This changes the expression of the gene, and so that can be designated as an allele. And please report about this clearly and the basis of this phenotype description within your paper. So there will be natural alleles that we will be reporting about, and these will be following the lowercase italic letter after the number. So for example, different alleles of RHTB1 would be called RHTB1A, RHTB1B. And the reference for this is Chinese Spring, which will be designated as the A allele. Dominant, semi-dominant, or codominant alleles should be designated with a capital letter, whereas recessive and null alleles should be designated with the lowercase letter, as shown here. And this is just a table which you can refer to in the manuscript that summarizes these particular guidelines. One thing that's sort of come about through new advances is naming alleles based on gene that have been developed through gene editing or through induced mutation, mutagenesis. And so induced mutants should basically follow a similar designation as what's occurred for haplotypes. Rather than use a H for haplotypes, we're recommending that you use an M for mutant. So for example, two different mutants for this agamus-like transcription factor, AGL12, should be called AGL12-A1 underscore M1 and or underscore M2. So this might, for example, be a study that's come from a study that studied the function of AGL12, and they developed two different mutant lines for this particular gene, and they're reporting about them in the paper, then they'd name them as such. Additional mutants for the same gene, additional mutants that are generated for the same gene in successive studies should then follow a sequential numbering system. So for example, if someone else investigated AGL12 in a successive paper, and they developed two different mutants, and they would call them underscore M3 and underscore M4. A similar recommendation is when you're describing a mutant that's been identified within the tilling population, then we ask that you include the identifier for that particular tilling line within your materials and methods. And details of the mutation should be provided when the mutation is first introduced. So for example, if it's a nonsense mutation that changes a valine to a methionine at position 63, then you report about that when you're introducing the particular mutant line. And so we recognize that this particular naming system might be a little bit cumbersome. And so if this particular allele that's been generated through a gene editing event or induced mutation is then used within breeding programs, then the allele designation that was described earlier with the lowercase letters can then be adopted. But this is only once it sort of becomes widely used. Another important area are SNPs. And in the interest of time, we'll just sort of quickly go over this, but there's a particular set of guidelines by which we're recommending that you name markers that are used to detect SNPs within genes that are protein coding, or if this is within an anonymous DNA sequence, then we recommend a particular set of guidelines that takes into account the genome by which it's been discovered, the version of that particular genome sequence, and then the location of that SNP. And the important part that we wish to point across with this is that this was discussed with the WEAK-CAP, the researchers that are part of the WEAK-CAP in the USA, And so we really hope that this will become a sort of consensus way for naming different markers that are based on SNPs. And there are other genetic topics that are discussed in this particular manuscript that I'd like to point out to you. So we also discuss guidelines for naming pseudogenes, gene complexes, particularly those that are involved in grain quality, QTL, acknowledgement of historical markers and how they're named, grain quality genes, and I've put pathogen or pest resistance genes here in green. And I'm not an expert in pathogen or pest resistance, and so I really don't want to sort of make a mistake in describing them to you, but they're quite clearly outlined within the manuscript, and I'd recommend that you talk to people such as Simon Kradiger or Bob McIntosh about the way that we name pathogen or pest resistance genes if you have any particular questions about those. A really important point, particularly from the people at CIMA who are authors on this paper, is that we would like any germplasm that's associated with a formally named gene to be made available internationally in a germplasm collection. This is really to help facilitate further genetic research in wheat, and I think this would be sort of a common goal that all researchers will have. But we just want to emphasize that, it's really important that once you discover a gene or an allele that you make this available to the international research community. And I guess finally, just some closing thoughts is that these guidelines are intended to be for the benefit of the wheat research community. So please use them, please consider them when you're naming your new baby wheat gene. We really want this to sort of benefit the wheat research community. We want to make it easier for you to know where to start with naming your gene. And we want to help improve that flow of information between academics and breeders and amongst the academic research community. We don't wish for them to be too prescriptive, but merely form a common reference point by which you can name genes. And we've tried our best through this process to accommodate historical conventions. We encourage you to support their use with your colleagues, particularly when it comes to their publications. So whether you're reading them within your department or when you're reviewing publications, or if you're acting as an editor, then we encourage you to use these guidelines. And indeed, we've already written to a number of journals to recommend that they include this in their guidelines to authors. We hope that it could be adopted by other cereal research communities, and we hope that it will benefit both academics and breeders. And I guess I'd really like to encourage you now to sort of go forth and discover and come up with some new and interesting names for your wheat genes. And finally, just to acknowledge all the people that have been involved with this, I've listed all the authors here, including where they do their research and their different funding bodies. And I'd particularly like to thank, just as a final note, the Wheat Initiative and a number of people who were part of the expert working groups within the Wheat Initiative who already had this particular idea running within their discussions. It's just they hadn't quite sort of got around to putting words down on paper. And they were really supportive when we approached them to formalize these guidelines. And so I'd really like to thank them with this effort. With that, I open to any questions.
Great, thank you so much, Scott, for this really great presentation. And also, I'd like to thank everybody, all the authors and collaborators for their effort to help whole wheat community to develop some kind of model that all the wheat community can utilize and use it, and both in academia as well as the breeders, that they can use it easily. So with that, I'd like to remind everybody that please just put your questions on the question board, and I'll go ahead and get started with the first question, Scott. And the question is how to nomenclature of the pleiotropic genes.
Of pleiotropic genes. So I guess that would be in reference to that first point about naming them according to the principal trait of interest. And so I would encourage you to sort of name it primarily. So particularly when you've discovered that locus, then you'll be naming it presumably based on a primary trait of interest. So say it affected, so as an example, we found a gene that affected spike development, but it also affected leaf architecture. But our primary trait of interest was spike development. So we named the gene originally based on its spike phenotype, even though we could see a leaf phenotype as well. And then to try and alleviate that potential confusion, we then name the gene based on the biochemical function of the transcription factor once we clone the gene. So it might be that you originally name it based on that primary trait, but then to reduce confusion, you then name it based on that biochemical function once the gene's discovered.
Great, thank you. So the second question, Scott, can you use the gene naming style in GWAS QTL naming?
Yeah, absolutely. So I guess the limitation with the GWAS would be that you're naming it based on a SNP originally, not perhaps you haven't yet cloned the gene, and so you'd be naming it like based on a position. So what we'd recommend there is that you give clear details about the SNP that came up with the best association with that particular phenotype, and then you could name it. You could give it the locus a name based on that primary trait of interest that you're investigating, and then you would then go through that sort of pipeline of naming it based on the biochemical function, perhaps once you clone the underlying gene.
Great, thank you. Another question is that, do you have any additional suggestions to help the adoption of these guidelines?
I mean, doing things like this talk is a great way to sort of broadcast them to the rest of the community. Twitter has been sort of fantastic about trying to publicize them when the manuscript was published, and we tried to get the international community involved. I guess it now comes down to more of that sort of community effort of just sharing it with each other when we see that someone perhaps doesn't name it in a way that's consistent with these guidelines. So I guess that's the thing, is that people become a lot more confident or a lot more secure in following a guidelines once it's sort of been published within a scientific journal. And so that's what we tried to do with this. Rather than it just being up on a website somewhere, we tried to sort of formalize it in a way so that people were more confident with following these guidelines. So it might take 10 years or something before these become like really uniformly used, but that's fine. We're happy with that.
Thank you. And another one is that nowadays a lot of publications are being reported on genome-wide identification, and unusually gene names are being used based on homology with model plants. If someone don't know the total number of the genes and their subfamilies in a specific gene family, what name should be used for a newly identified gene?
So I think that's where we start at the number 1, and then we go from there as things evolve. We appreciate that there's still sort of hidden secrets within the wheat genome, and that things will evolve and the literature will evolve accordingly. And so we recommend that, for example, if you find a gene that might be like, imagine, so for example, a GA20 oxidase, there's at least 3 GA20 oxidases out there that control growth in wheat. And if you're the first person to discover GA20 oxidase in wheat, then you would name it GA20-OX1. And then any successive discoveries that were made of GA20-OX after that would be then given the successive numbers 2 or 3. And I guess that's where what we've also noticed within the literature is that people are quite happy to name genes without necessarily having gone and done the phylogeny or check to see if there are extra copies. So it's really not that hard these days to do a quick blast search and see if there are other copies of your gene in the wheat genome. So it might just be like a supplementary figure or something like that. But if you can do a little bit of that investigative work to see if there are other copies, then it might help you out with your own understanding of that particular gene, and also contribute to other people's discoveries. So yeah, so start at 1 and increase the numbers from there. But yeah, if you can do a little bit of extra work investigating that gene and do some phylogeny, then I think it would be really helpful to a lot of us.
Thank you. And based on the question dashboard, there's not many. And I just want to ask one question, Scott. And I know it's too late for you, but I just wanted to ask still, what is the next then? Are you planning to expand that the whole wheat community as well as that the new genes or non-genic regions as well, like non-coding RNAs, or what are the next step and what is your plan?
Yeah, that's a really good point, because I guess to start off with, we were just conscious of the fact, like I talked to Bob McIntosh, who was like the curator of the wheat gene catalog, and we originally talked at the meeting in Austria in 2013, I think it was, and he was well and truly into retirement then. He was happy to sort of pass on the stewardship of the wheat gene catalog, but since then things just grew and grew and grew, and it just became something too big for any one person to sort of handle. So yeah, we haven't really talked about that yet, but it is a really good thought, is, you know, what does come next. I think we'll sort of be driven a little bit by the discoveries that are made and how many different cultivars are sequenced, and what, you know, whether eventually a pan genome becomes like the reference rather than Chinese spring becoming the reference. There are ways, there's sort of conventions out there within the plant community in terms of how to name microRNAs and long non-coding RNAs and short interfering RNAs. So I guess a lot of the similar principles will be maintained with what we've already put in these guidelines, but it'll ultimately be driven by where the discoveries come from. And I guess that's quite exciting in a way.
Great. One thing that I have to add as well, that when you naming the genes, if it is mutated, you put the lowercase m, and when you edit the genes, and let's say we have edited lines, and you are going to put, let's say, E for the gene naming, E instead of M, would that work? Yeah, E would be, this is just a suggestion.
Yes, edited instead of me.
Instead of M, because that is going to confuse a lot of people, and instead of M, because we have tilling mutant lines, and we can put M, but edited lines, we should, this is a suggestion. E is going to be a good indication for,
Yeah, that's a really good point. Thank you. That's an awesome suggestion. Just one last thing that I forgot to say in one of my last slides is I do have a PhD position in my lab at the moment. This is sort of like a personal plug, but if any of you want to come and investigate genes and name them in my lab, then I have a PhD position open at the moment. It's even open for international students. So yeah, if that's something that you're curious about, then please write to me and I'd be happy to talk to you about it more.
Great, thank you so much. And I'd like to thank you really, Scott, for this great presentation, and as well as all the efforts for the gene naming. And I'd like to thank everyone who stayed with us, and hope to see you next webinar. Thank you so much.
Thanks very much, Hikmet.
Thank you.